This video is part of the iGEM Academy series, a beginner's guide. This tutorial shows how to prepare a variety of solutions, including sodium chloride solution, glycerol solution, calcium chloride solution, TBE buffer, LB medium, and an LB Agar plate containing antibiotics. First, the sodium chloride solution will be prepared. This solution is used during experiments in order to prevent osmosis, which damages the cells. In this example, 10 milliliters of 0.9% sodium chloride solution is prepared. For this, sodium chloride salt and doubly distilled water, which will be referred to simply as water in this video, are needed. To make up 10 milliliters of 0.9% sodium chloride solution, 0.09 grams of sodium chloride salt is weighed out. The appropriate amount of salt is then added to a beaker. It is good practice to take the pipette out of its packaging only when you are ready to pipette. Water is now added to dissolve the salt. In this case, 10 milliliters of water is added. 0.9% sodium chloride solution has now been made. Next, a 50% glycerol solution is prepared. This solution is used for making cell glycerol stocks out of bacterial strains. The presence of glycerol in the stocks will prevent damage to the cell membrane of the bacteria. To make a glycerol solution, the following are needed: glycerol and water. In this example, 20 milliliters of 50% glycerol solution is made by adding 10 milliliters of water to a beaker. 10 milliliters of glycerol is then added to the water. The solution is then covered with aluminium foil and autoclaved for 20 minutes. A 50% glycerol solution has now been made. Next, a 1 molar calcium chloride solution is prepared. Calcium chloride is used to make E. coli cells competent. In this video, 10 milliliters of 1 molar calcium chloride solution is prepared. To make calcium chloride solution, the following are needed: calcium chloride and water. 10 milliliters of water is added to a beaker. To make 10 milliliters of 1 molar calcium chloride, 1.11 grams of calcium chloride is weighed out and added to the beaker that contains the water. The beaker is swirled until calcium chloride fully dissolves. Calcium chloride solution has now been made. Make sure to autoclave for 20 minutes before use in the lab. Now, 10x TBE buffer is prepared. TBE stands for tris, boric acid, and EDTA. EDTA chelates magnesium. This inhibits enzymatic degradation of nucleic acid and gives sharper bands in gels. For the TBE buffer, tris base, boric acid, EDTA, and water are needed. 80 milliliters of water is added to a beaker. 10.78 grams of tris is weighed out and added to the beaker that contains the water. Tris is dissolved in water using a magnetic stirrer. 5.5 grams of boric acid is weighed out and added to the same beaker that contained tris. Boric acid is allowed to dissolve. 0.7 grams of EDTA is weighed out and added to the same beaker. 20 milliliters of water is now added to make up the final volume of 100 milliliters. 50 milliliters of this solution that has now been made up is transferred to a properly labeled glass bottle. The solution is diluted times 10 with water so that the final volume in the bottle is 500 milliliters. TBE buffer has now been made. It is stored at room temperature in the glass bottle. Now, lysogeny broth, or LB for short, is prepared. This medium is frequently used for bacterial growth. For LB medium, the following are required: sodium chloride salt, tryptone enzymatic digest from casein, yeast extract, water, and 5 molar sodium hydroxide solution. First, a 5 molar sodium hydroxide solution is prepared in the following way. 0.02 grams of sodium hydroxide is weighed out and transferred to an Eppendorf tube. The salt is dissolved in 100 microliters of water. To make 500 milliliters of LB, the following are added into an empty glass bottle. 5 grams sodium chloride, 5 grams tryptone, and 2.5 grams yeast extract. Water is added to the bottle containing the salts. The glass bottle is shaken to allow the salts to dissolve. The pH is adjusted with 50 microliters of previously prepared 5 molar sodium hydroxide solution. Water is added to make up a final volume of 500 milliliters. The cap on the bottle is loosely closed. The top is then loosely covered with aluminium foil. The solution is autoclaved for 20 minutes. LB solution can be stored at room temperature. LB agar plates containing specific antibiotics allow resistant selection of bacteria. The following are needed to prepare agar plates: freshly prepared LB medium that hasn't been autoclaved, agar, an antibiotic of choice at the right working concentration. Ampicillin was used in this video. To a glass bottle, the following are added: 300 milliliters LB prepared fresh that hasn't been autoclaved, and 4.5 grams of agar. The bottle is shaken. The bottle cap is loosened and the top is covered with aluminium foil. The mixture is autoclaved for 20 minutes. The mixture is allowed to cool so that the bottle is touchable. 300 microliters of the antibiotic is now added. The antibiotic used in this video is ampicillin. About 20 milliliters of the made-up mixture is poured into each petri dish. The petri dishes are kept close to the Bunsen burner flame to prevent contamination. The plates are left to solidify for about an hour. Solidified plates are then stored in a fridge at 4 degrees Celsius. Thank you for watching. This video is brought to you by the TCD iGEM Team 2015. To go directly to the next video in the series, A Beginner's Guide, please click on the link provided.